Good day, this is Professor Will, CBMG PhD, and today is Saturday, January 15th, 2022. It is 7:09 and 39 seconds, p.m. Eastern Standard Time. Today's research, cell death discovered. So let's hop to it here. Deadly combination, new direct trigger for cell death discovered. Date January 13th, 2022. Source, University of Columbia. Understanding the basic mechanisms that lead to cell death, apoptosis, is essential for the development of therapies for cancer and other diseases. The scientists have now discovered a protein interaction that directly triggers cell death. It involves a direct physical interplay between the two proteins, BAX and DRP1. DRP1 can serve as a direct cell activator by binding a BAX without the need for other cell death triggers. And this finding could lead to the development of new cell death regulators for cancer therapies. For example, the article, DRP1 interacts directly with BAX to induce its activation and apoptosis. It is known that the so-called apoptotic enforcer, protein BAX, encounters DRP1 in the cell at the mitochondrial membrane. The latter is a dynamine-like protein that plays a critical role in mitochondrial division. However, the functional implications of their interaction and the contribution of DRP1 to apoptosis have been highly controversial. BAX is a key protein in the pathway to cell death. Understanding the mechanism of action of BAX is critical for therapeutic regulation of apoptosis. And using super-resolution confocal fluorescence, microscopy, and biochemical as well as biophysical methods and model membrane systems, the research team was able to demonstrate the direct interaction of two proteins in dying cells. In addition, using a system that artificially brings the two proteins together, they investigated the functional consequences of the interaction of BAX and DRP1. When scientists force the interaction of the two proteins, they move from the cytoplasm to the mitochondria, where the protein complex triggers a reorganization of the mitochondria. And this leads to pores in the membrane, the contents of the mitochondria, into the cell plasma, which ultimately leads to cell death. By combining methods such as a dimerization, dependent fluorescence technique, cross-linked mass spectrometry, and the analysis of different protein pieces, the interaction surface can also be identified for the first time. DRP1 binds to the front end and terminus of the amino acid chain of BAX, which is shown to be a regulatory region for BAX activity. It was impressive to see that the cells start to die just by forcing the interaction between BAX and DRP1 without the need for another death trigger. It is great that we now know that DRP1 can act as a direct apoptosis activator, which for the first time gives functional significance to the connection between the two proteins. And this could pave the way for the development of new BAX regulators for therapeutic applications. Good day, this is Professor Will, CBMG PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.